In the clinic, we treat a lot of patients with kidney disease. These patients have kidneys that do not work well anymore. And the reason these kidneys do not work anymore often is the cause is high blood pressure or the diabetes, which is destroying the kidneys. Now, in case of high blood pressure, it's also the kidneys that regulates the high blood pressure. But if the blood pressure is too high, it destroys the kidneys. In the case of diabetes, which is about half of all patients in which the kidney doesn't work anymore, which are on dialysis, they have diabetes. It's either that the beta cells in the pancreas do not secrete enough insulin, or the other reason is that the insulin cannot work anymore in the periphery. These are the two reasons. In our lab, we are studying sodium proton exchangers. These are little enzymes that are sitting on the plasma membrane of cells and are extruding sodium in exchange of protons. And we are studying a peculiar exchanger which is important in the kidney for blood pressure regulation and in the endocrine pancreas for the secretion of insulin.
Here at the Department of Chemistry and Biochemistry, we try to understand these sodium proton antiporters like NHA2 and its isoforms at the more molecular level. This means we try to understand the basic molecular mechanisms and how this affects its function in the cell. So one aspect to do so is to find small molecules that inhibit or activate these enzymes, so-called modulators.
Our main aim is to test the modulators that were found by our collaborators in vivo on the insulin and glucose level of primary cells as well as animals.
Mainly we screen small compound library to get modulators of the protein. So we use a ligand-based virtual screening approach using tools developed in our lab. From this library, we screen it in a yeast assay developed by the Daniel Fuster group, and from its discover, we then optimize them by chemical synthesis.
Additionally, we also work with the purified enzymes. To this end, we take the human DNA and express it in yeast systems. We purify then the enzymes and put it back into a synthetic membrane lipid bilayer. We then test the activity of NHA2 and its isoforms by means of fluorescent-based assays.
Our project is really interdisciplinary. Our goal is to develop and search for new molecules that could be used in the future as drugs to treat high blood pressure, diabetes and bone loss in our patients.